Hi students, today I am going to discuss about gene interaction. So in the previous module we have seen the Mendelian theory, okay, the law of inheritance. So we have seen the law of dominance, law of segregation and law of independent assortments with the examples of monohybrid cross and dihybrid cross, okay. So according to the law of dominance, when two pure breed are crossed, okay, they have one generation, only one of the parent traits appear phenotypically. Okay, the appeared traits or that expressed character is known as a dominant, which is not able to be expressed, and it is known as a recessive. However, the recessive trait expressed in F2 generations when F1 is allowed for the self-pollinations. Today we are going to discuss about certain laws, certain concepts which are exception to the law of dominance. Examples: incomplete dominance, codominance, okay, over-dominance. These are all exceptions to the law of dominance. How it is exception to the law of dominance and what actually it tells about the gene interactions. Okay, so the gene interaction, what is the gene interaction? The interaction between the two genes or more genes. Okay, when they interact, the result will be the phenotype modification or the modified phenotype. Okay, the result of the gene interaction will be the modified phenotype. The most of the characters of the organism is 100% the result of gene interactions. The chemical structure, the structural characters and chemical composition of all these phenotypes is the result of interaction of the gene. Okay, gene interactions are classified into intragenic interaction and intergenic interaction. What is an intergenic interactions? So this interaction is between the alleles, the two alleles of the same gene, okay. So that's an intragenic interactions. So the interaction happens between the two alleles of the same gene, okay. But the intergenic interaction, it happens between the interaction happens between the two non-allelic genes. Okay, let's move on to the definition of gene interaction. So gene interaction is the modified function of the normal gene phenotypically. Okay, so the modification of the normal gene which express the phenotypes due to its alleles are the non-allelic interactions. Okay, so under the intragenic interactions, so we can see different things. Dominant relationship. Okay, so in case of dominant relationships, see that what are the things will come under the dominant relationship? Complete dominance we have seen in the monohybrid. So tall plants is crossed with the dwarf plants. So what happens? The F1 hybrids will express one of the parent trait, that is a tallness. Okay, so that is a complete dominance. One allele is dominant over the other alleles of the gene. Okay, next one is an incomplete dominance. So incomplete dominance, it's a phenomenon which is first observed in the plants mirabilis, jalapa, that is an 4 o'clock plants. Okay, so that's 4 o'clock plants, the mirabilis jalapa having the red flowers as well as the white flowers. Okay, so some parents of the mirabilis jalapa have the red flowers, some plants of the mirabilis jalapa will have white flowers. Okay, who has observed this phenomenon? That's the correns. So he is the person observed that incomplete dominance in mirabilis jalapa, that is an 4 o'clock plant. Okay, so what happens in incomplete dominance? So one dominant allele does not express completely itself. Dominant allele does not express completely itself. Okay, so that is 50% can be expressed in the heterozygote conditions. 50% of dominant allele expressed in the heterozygote conditions. So look at this the flow chart. So you will identify. So this is an parent generations of that mirabilis jalapa. So it is having the alleles, that is an R1. So R1 gene. So R1 gene is responsible to encode the functional red pigmentation formation. Okay, it encode the formation of the red pigments. Okay, R2 is a defective and muted. R2 is a defective and muted. Okay, so the red flowers is of the mirabilis jalapa having the alleles R1. Okay, R1, that's a dominant allele. Okay, when it's crossed with the white flowers of mirabilis jalapa, which is having the R2 allele, which is a defective or muted allele. Okay, which is a defective and muted allele. So red flowers of the mirabilis jalapa is crossed with the white flowers of the mirabilis jalapa. That in F1 generations, so we are able to see a pink color flowers. Pink color flowers. Look at this. We have taken red flowers and white flowers. We crossed these two things. But in F1 hybrids, we are able to get pink color flowers. How it is possible? So in this, you see that alleles R1 expresses 50%. Okay, so R1 is a functional gene, so thereby it expresses itself 50%. So it is not expressing completely in the heterozygote conditions. Only 50% of the functional gene is expressed and forms only 50% of red pigments. Only 50% of red pigments. So that's why we get it in pink colors. Okay, so look at that. So thereby we can understand. So one dominant allele does not express completely itself in the heterozygous conditions. Clear? So from this we can conclude incomplete dominance. So in incomplete dominance, there is no blending of gene. There is no blending of gene occurs. Okay? So that blending inheritance theory is disproved. Blending inheritance theory is disproved. So intermediate color is formed. Intermediate color is formed because of the dominant allele expressions in 50% ratio. Okay, do you understand? When this F1 is allowed for the self pollination, when this F1 is allowed for the self pollination, and we are able to get red flowers and white flowers and pink flowers. Okay, so and F1 is self pollinated. F1 is self pollinated. And F1 is self pollinated, we are able to get red flowers, pink colors as well as the white. Okay, this is an dominant allele, so thereby it shows the red color. Okay, and this is following the law of segregation, randomly reunites and forms a new trait. So here it shows the pink color because the R1 gene expresses only 50%, 50. Okay, so it is not completely expressing itself, only partially expressing itself. So thereby it forms the intermediate color, that is a pink color. The same way, this is also pink color, and this is an recessive, muted, defective alleles. So thereby forms an white flower. Okay. So what will be the phenotypic ratio in the F2 generations in this case? This is an F2 generation. What will be the phenotypic ratios? So look at this, the pure homozygous red color. Okay, so we are having that 1. Pure homozygous red is 1. Okay, then we are having the intermediate form. How many intermediate forms are there? 1 and 2. So 2 intermediate forms, pink color. Then again recessive, defective 1, that is an 1. So the phenotype as well as genotype ratio of F2 generation in incomplete dominance is 1:2:1. 1:2:1. So 1 pure red colors and 2 intermediate pink colors and another one is white colors. Okay, got it? So do you understand what is an incomplete dominance? So incomplete dominance la enna nadukuduna நம்ம mirabilis jalapa plant எடுத்துட்டு அதை வந்து நம்ம கிராசிங் பண்ணும்போது அந்த 2 பேரண்ட்ஸ் 2 பியூர் பேரண்ட் ப்ரீடை வந்து கிராஸ் பண்ணணும். ஒரு ப்ரீடு வந்து ரெட் ஃப்ளார்ஸ் இருக்கு, ஒரு ரெட் ஃப்ளார்ஸ் இருக்கிற பிளான்ட், இன்னொரு ப்ரீடு வந்து ஒயிட் ஃப்ளார்ஸ் இருக்கிற பிளான்ட். இந்த ரெண்டு ப்ரீடையும் கிராஸ் பண்ணும்போது F1ல அக்கார்டிங் டு லா ஆஃப் டாமினன்ஸ் பொறுத்த வரைக்கும் ஏதாவது ஒரு பேரண்ட்டுடைய ட்ரேட் தான் அந்த F1 ஹைப்ரிட்ல அப்பியர் ஆகணும். பட் இந்த நம்ம இங்க ரெட் ஃப்ளார்ஸையும் ஒயிட் ஃப்ளார்ஸையும் எடுத்து கிராஸ் பண்ணும்போது F1 ஹைப்ரிட்ல நமக்கு பிங்க் கலர் கிடைக்குது. ஓகே, ஸோ பிங்க் கலர் கிடைக்குது. இதுலேருந்து நமக்கு என்ன தெரியுதுன்னா, ஒரு அல்லீல் வந்து டாமினன்ட் அல்லீல் கம்ப்ளீட்டாக எக்ஸ்பிரஸ் ஆகாமல் பார்ஷியலாக எக்ஸ்பிரஸ் ஆகுது. ஓகே, இது அது கம்ப்ளீட்டாக ஹெட்ரோசைகேட்டில் கம்ப்ளீட்டாக எக்ஸ்பிரஸ் ஆகாமல் பார்ஷியலாக எக்ஸ்பிரஸ் ஆகிறதால 50% கலர் ஃபார்மேஷன் தான் நடக்குது. அந்த பிகாஸ் ஆஃப் 50% கலர் ஃபார்மேஷனால தான் நமக்கு பிங்க் கலர் கிடைக்குது. ஓகே, R1 ஜீன் என்றது இட்ஸ் என்கோட், அதாவது ரெட் பிக்மெண்ட் உருவாகிறதுக்கு ஒரே புரோட்டீனஸ் சிந்தசிஸ் பண்ணுறது. R2 என்றது டிஃபெக்டிவ் ஜீன், அதாவது மியூட்டட் ஜீன். ஸோ இதனால் வந்து ரெட் பிக்மெண்டேஷனை உருவாக்க முடியாது. ஸோ இட்ஸ் அண்ட் ஒயிட் இன் கலர்ஸ், வைல்ட் ஒன். ஓகே, இந்த ரெண்டுமே சேரும்போது ஆட்டோமேட்டிக்லி வி வில் கெட் இட் அண்ட் ஹெட்ரோசைகட்ஸ் பிங்க் கலர். அப்போ இந்த ஹெட்ரோசைகட்ஸை நம்ம திரும்பி செல்ஃப் பாலினேஷனுக்கு அலோவ் பண்ணால் வாட் ஹேப்பன்ஸ், நமக்கு ரெட் கலரும் கிடைக்குது, பிங்க் கலரும் கிடைக்குது, ஒயிட் கலரும் கிடைக்குது. ஸோ அது ரெட் கலர் ஹோமோசைகஸ் வந்து ஒரு இது, அடுத்தது 2 பிங்க் கலர் இன்டர்மீடியேட் கிடைக்குது, ஒரு ஒயிட் ஃப்ளார் கிடைக்குது. அப்போ F2 ஜென்ரேஷனில் ஃபீனோடைப்பும் ஜீனோடைப் ரேஷியோ வந்து 1:2:1 இந்த விகிதத்தில் தான் இருக்கும். ஓகே, ஸோ லெட்ஸ் மூவ் ஆன் டு நெக்ஸ்ட் ஒன் இஸ் இன் கோடாமினன்ஸ். ஸோ இந்த கோடாமினன்ஸில் எப்படின்னா 2 அல்லீலும் எக்ஸ்பிரஸ் ஆகும், ஈக்குவலாக எக்ஸ்பிரஸ் ஆகும். In heterozygous condition, heterozygous condition the alleles, both the alleles express itself equally. 2 அல்லீலும் ஈக்குவலா heterozygous conditionல எக்ஸ்பிரஸ் ஆகும். அதே போல தான் F2 ஜெனரேஷன்ல அதோட phenotype genotype ரேஷியோ வந்து 1:2:1 ரேஷியோல தான் இருக்கும். ஓகே, வாட் இஸ் தி கோடாமினன்ஸ்? Codominance. What is codominance? What is codominance? Both the alleles of a gene express equally in heterozygous, in heterozygote in heterozygotes. So both the alleles, some in case of ABO blood group for A blood group, so the allele will be IA அப்படி இருக்கலாம், or IAIO. Okay, both the things will be expressed. Example, AB blood group la allele வந்து IB gene, IB gene. So both the things will give A group, B group, AB group. Do you understand what I am saying? Codominance theory la enna, both the alleles of the gene express equally in heterozygote condition. Do you understand? Okay, so this is an ABO blood group is an best example. ABO blood group is the best examples. Okay, so another thing is that red and white color, white flowers of the gamelio. Okay, red and white colors of the gamelio plants. Okay, so next thing you will identify sickle cell hemoglobin. The sickle cell hemoglobin also shows the codominance. Got it? Clear? So what in codominance? Both the alleles are expressing equally and with an equal proportion in the heterozygous condition. So thereby it shows the two things. Okay, Ronan coat of the cattle. Ronan coat of the cattle. If you take the dog also, okay, one dog is in white colors. Okay, example that male dog is in white, having white fur. Female dog is having that black fur. So when they undergoes that mating process and the female dog give birth to the puppies. Okay, this puppy, some of the puppies will be having pure white, some of the puppies will be black color, some puppies will have both the colors, black patches, white or white patches with black. So all this is this. The proper they will be expressed individually without any disturbance. Got it? Clear? That will be that good example to remember. The Ronan coat of a cattle, okay, or then dog. Or in case of plant, you will identify the gamelio plants. Okay. So how in plants we can find out the particular gene, the particular alleles is expressed equally. So that is because the electrophoresis method. When we follow the electrophoresis method, we are able to identify the protein synthesis, this protein. So by separating out the proteins, we are able to understand which allele, which gene is expressing which color, which type of protein. So both the both the alleles are expressing equally to give particular protein synthesis. Do you understand? Thank you for watching my channel. Okay, so in next modules we will discuss about the lethal genes.